Okay, can somebody confirm you can hear me?
We can hear you.
Great. Let me start by saying that I have graded the lab. Let me see which one. I have graded lab 3. So you can make corrections by this Saturday if you did not get 100%. I have not graded lab 4 yet, and I don't know if I'll get that finished tonight. If I do not get it finished tonight, I'll extend the until date, meaning you'll have another week to get it turned in. But if I do finish it tonight, then it will be due, the second grading will be due on Saturday. Any question about any of that? There was not a lecture worksheet, which is a little unusual. It's probably because lecture 3, we're only part way through it, and there's only one lecture worksheet, so it'll be due this week. And I think lecture 4 will also be due this week. Let me check on that right now. No, I'm not finding that now. Well, let me edit it and then I'll find it. Let's skip it for now. All right, any questions about anything? If not, let me share my screen, get out of the grade book, I guess. Let me share my screen. Um, and we are, well, let me state today is October 15th. I'm just checking to make sure I've got the lecture 2 available. It is. All right, great. Today is October 15th. We should be finishing chapter 3. There is a quiz this Wednesday covering labs 3 and 4, chapter 3, and the end of chapter 2. Any questions about any of that? There will be a lab. There will be a lab today. All right, let's move on to chapter 3. There it is. So I have that we were here. Is this correct? That we had talked about gram staining?
Yes.
All right. I don't know why it says Monday, Wednesday. Just check to make sure I don't have another one. Slide 41. Or had we talked about the basis of gram staining? Can anyone tell me, did we end at slide 41 or slide 47? This is slide 47. Let me go back to slide 41. Anyone?
We ended at slide 41.
All right. So, repeating slide 41 just briefly, the gram staining. After you stain with safranin, the color of the gram-positive cells will be purple. The color of the gram-negative cells will be pink. You then blot the slide dry with bibulous paper. Bibulous paper is a very thick paper, so pretend this is the slide. It's very thick paper. It's almost like thin cardboard. You get the slide like this, and then you just blot it dry. And that means you press down on it. You do not take the slide and wipe it on the bibulous paper. You don't wipe the bibulous paper on the slide either, because that can wipe off the cells. That will remove enough of the water to allow you to see the cells under the microscope, even if you're using oil. All right, so the gram-positive cells are purple, the gram-positive negative cells are pink. Why? What is the basis of the differential staining? The differences are related to the differences in the cell wall structure. The gram-positive bacteria have a thicker cell wall with a thick peptidoglycan layer. We'll talk more about peptidoglycan, I think, in our next lesson. But it has a very thick peptidoglycan layer, and the crystal violet iodine complex cannot wash out of that thick peptidoglycan layer. The gram-negative bacteria have a thinner cell wall with a thinner peptidoglycan layer. And the crystal violet iodine complex can wash out of the thinner peptidoglycan layer. The gram-negative bacteria also have a lipopolysaccharide layer. It's part of the cell wall. We call it the outer membrane of the cell wall. And in the decolorizing step, that destroys the gram-negative outer membrane of the cell wall. I thought I had shown you this picture, but let me go over it again. And that is the gram-positive cell wall has a very thick cell wall, which is made up of layers of peptidoglycan. There's only 3 layers shown here, but in reality, there's at least 20 layers. There may be 100 or more layers of peptidoglycan in the gram-positive cell wall. In the gram-negative cell wall, it's a much, oops, too far. Let me go back. In the gram-negative cell wall, it's a much thinner peptidoglycan layer, and then it has this lipid bilayer on the outer side of the cell wall. We call that the outer membrane of the cell wall. This is only showing you 1 layer of peptidoglycan. In reality, the gram-negative cells have at least 3 layers of peptidoglycan, and they can go up to about 20 layers of peptidoglycan, but not beyond that. Any question about any of that? So the point being, the cell wall of peptidoglycan is much thinner in the gram-negative cells than in the gram-positive cells. Let me shrink that down so I can read it. So the crystal violet iodine complex in the gram-negative cells is washed out in the decolorizing step. Therefore, the cells become colorless. The gram-positive cells retain the crystal violet iodine complex, and the cells remain purple. And I do remember, I did show you this picture showing you the different gram-negative and gram-positive cells. So we have discussed this. All right, the gram stain is not entirely trouble-free. Like, for example, what happens when you get a slide looking like that? Those cells look much lighter than these cells there. These are obviously dark purple. And how do you read this slide? Well, you skip the regions of the slide that are bad. And then you read the cells where the cells are good. Wherever there's a very dark purple spot like that, that's actually a crystal of crystal violet. Crystal violet, especially if it's not freshly made, will start crystallizing in the bottle. And if you use the bottom of the bottle, you'll get lots of crystals coming out onto your slide. You do not want to read the cells around the crystal because, let me blow this up a little bit, it'll show it. I've seen gram-negative cells, these are actually gram-positive cells here, but I've seen gram-negative cells will be staining dark purple around the crystal of crystal violet. What happens is the crystal violet comes out of this crystal and it will then stain the cells around it purple. And then the cells further out will be staining pink. So you don't read the slide where there are crystals of crystal violet on the slide. You also don't want to read the cells where the cells are clumping. These aren't a clump, that's actually a crystal. That would be sort of a clump. You want to read the cells where the cells are single layer thick. And the gram stain is a little tricky, which will become apparent to you when you're working on the unknown project, especially if you were going to be in the lab actually doing the gram stain yourself. We, of course, are going to do this online, so you will not be doing the gram stain. And I won't send you a slide where it's tricky and you have to figure out how to read the slide. I'll give you a good slide of the gram stain. Most consistent results in the gram stain are obtained when you use the gram stain on young growing bacteria. If your cells are older than 2 days old, you should not do the gram stain. You should take your cells and grow them up for a day or two, and then use those freshly grown cells to do the gram stain. The gram stain does not work, or at least does not work well, if your cells are not less than 2 days old. If they're older than 2 days old, the gram stain could give you false results. Gram stain is also not universally applicable. Some bacterial stain poorly, if at all, and it's mostly the mycobacterium which follow this staining poorly. Every time I've used stained mycobacterium, the cells do stain gram positive. However, they stain lighter than normal gram positive cells. And that's because the mycobacterium have a lot of mycolic acid in their cell wall that prevents the crystal violet iodine from coming into the cell and then staining the cell wall. And like I said, the results are not always as clear-cut as the theory suggests. If you were to do the gram stain, it would be very apparent to you. You'd be pulling out your hair sometimes when you do the gram stain. It takes a little practice to do the gram stain and then get it to work well. I've got another slide here. Let me come back to this one. Nope, I don't have it here. All right. Like I said, the main thing to do is to have freshly grown cells less than 2 days old. You also want your cells about a single layer thick on the slide. If there's too many layers, it'll be hard for the stains to get down into the bottom layers. And then it'll also be hard to decolorize the bottom layers of cell if the decolorization have to get through several layers and then wash it out of several layers thick. So you want cells that are freshly grown and cells that are very thin, meaning a bacterial smear that's very thin, ideally 1 layer thick. The structure of the gram-positive and gram-negative cell walls not only affects the staining, it also is related to how they respond to antibiotic. Gram-positive bacteria tend to be killed easily with penicillin G, as well as cephalosporin and many detergents. The gram-negative bacteria are more resistant to these antimicrobials. Like penicillin G cannot get through the outer membrane of the gram-negative cell. So penicillin G does not kill gram-negative cells. And these gram-negative cells are resistant to some detergents, cephalosporin, and then penicillin G and a few other members of the penicillin family. Any question about any of that? That doesn't mean gram-negative cells are resistant to all antibiotics. You just need an antibiotic that works on gram-negative cells, like streptomycin. Any question about any of that? All right. Let me also state that the archaea, which have different cell walls, they don't have peptidoglycan in their cell wall, but the archaea bacteria, or archaea, do stain gram-positive and gram-negative. It turns out that the gram-negative archaea have a thinner cell wall, and the crystal violet iodine complex washes out of that thinner cell wall. The gram-positive archaea have a thicker cell wall, and the crystal violet iodine complex does not wash out of that thicker cell wall. So the archaea stain, I guess, similar to the bacteria with the gram stain. The difference is the archaea do not have peptidoglycan in their cell wall. I think it's pseudomurine. You don't need to know that, is what they have instead of peptidoglycan. I won't quiz you on that because we're not going to be quizzing you on the archaea other than obviously it's a domain. All right, let's talk briefly about the capsule stain. It is one of the special stains that stains a capsule. Remember, a capsule is a glycocalyx that you can see under the light microscope. It has molecules in it other than water. The slime layer, really, mostly what it is just water. And that's why it doesn't show up with the light microscope. You can see the slime layer with an electron microscope. But the capsule can be seen with the light microscope. It has more molecules in it than just water. And it is more rigid too than the slime layer. The capsule stain, I'm not going to quiz you on this, but you need to know it because you may use the capsule stain for your unknown project. The capsule is a glycocalyx that you can see in the light microscope, and it's a layer secreted by the bacteria cells. So it's external to the cell wall, so we call it external to the cell, but it's made by the cell, and it essentially offers protection to the cell, particularly protection from a white blood cell, which would phagocytize the bacteria. If the bacteria has a capsule, it will escape phagocytosis by white blood cells. It can also help protect the cell from desiccation and some other things, like help protect it against antibodies and the immune system. Anyways, here's the capsule stain. It stains the background of the slide and the cell. This is probably a differential capsule stain using two dyes, one to stain the background, one to stain the cell. This is a negative stain just using one dye, India ink, which stains the background very dark and then lightly stains the cell, sort of a light gray, and the capsule remains clear in both capsule stains. Any question about any of that? All right. If there's no questions, let's begin chapter 4. All right, chapter 4 is the functional anatomy of prokaryote and eukaryotic cells. We will talk about the functional anatomy of prokaryotic cells. Eukaryotic cells, I almost never discuss. And that's because if I run out of time, you should have had the functional anatomy of eukaryotic cells in Biology 160. And so I will make the slide available to you if you need to review it, but you should have had the functional anatomy of eukaryotic cells, and so I will probably not discuss it. The exception is if we have lots of extra time. I will talk about one thing about the anatomy of eukaryotic cells, and that is the endosymbiotic theory. I will talk about that. All right, so this slide is showing you the major goals and the rough outline of chapter 4. Know the terms. What's a conchus, a bacillus, a spirilla? What is a strepto, like a streptococcus, a staphylococcus, a diplococcus? What's a tetrad, a Sarcenia? What's a vibrio? What's a spirochete? What is a glycocalyx? What is the flagella? What is an axial filament, also called an endoflagella? What is a fimbriae? What is a pili? What is peptidoglycan? We'll talk about peptidoglycan a lot here. What is an endotoxin? What is selective permeability? What is diffusion? Facilitated diffusion, osmosis, active transport. What is tonicity? What is a plasmid? What are inclusions? Endospores. And then lastly, we'll talk about the endosymbiotic theory to end this lesson. Two, you should know how gram-positive and gram-negative cell walls differ. And then lastly, 3, describe the importance of taxis in bacteria. Any questions about this overview and the major goals of slide to chapter 4? Remember, take a look at the study guide if you want to get all of the goals that I call objectives for the lesson. So this slide is showing you the difference between prokaryotic cells and eukaryotic cells. Prokaryote actually is Greek for meaning pre-nucleus. Literally, it means pre-kernel, and the kernel is used for the nucleus. And then eukaryote is Greek for true nucleus or true kernel. Prokaryotes generally have one circular chromosome. So the chromosome, I'm kind of trying to draw it here, is in a circle, and that means the place where it begins is where the place where the chromosome ends. The chromosomes are not in a nucleus. Eukaryotes have a linear chromosome, meaning all the chromosomes are linear, and the chromosomes are in a nucleus. The X chromosome is a linear chromosome, and it is a beginning place, which differs in the ending place. Any question about any of that? For bacteria, the DNA is not associated with histones. There are no histones, which is a protein. There are no histone proteins in bacteria. For eukaryotes, the DNA is wrapped around a histone protein. So histones are actually the main protein in the eukaryote DNA. Now, DNA of prokaryotes does have other proteins, and the archaea even has a histone protein, but we're not going to talk about the archaea. We're only going to talk about the bacteria and the eukaryotes. The prokaryotes have no organelles. By organelle, I mean a membrane-bound organelle, something like the Golgi complex, or the nucleus, or the vesicle. And the eukaryotes do have membrane-bound organelles. There is such a thing as a non-membrane-bound organelle, and I don't usually use that terminology. So when I use the word organelle, I'll mean a membrane-bound organelle. A non-membrane-bound organelle would be something like a ribosome or a, what is that called? Centrosome, something that does not have a membrane around it. And ribosomes, of course, are found in bacteria. Peptidoglycan is found in the cell walls of bacteria and bacteria only, meaning it tends not to be found in archaea. Peptidoglycan is not found in the cell walls of eukaryotes. So this is one area where bacteria differ from eukaryotes, as well as archaea. And then the prokaryotes, their cells divide by binary fission, where the cell literally splits down the middle in two. And eukaryote cells divide by mitosis, which we're not going to talk about. You should have had that in Biology 160. So in this chapter, we're going to talk about prokaryotes, discussing the cell shape and the cellular growth arrangement. We'll then talk about the cell structures and the movement of material across membranes. If we have time, we'll talk about eukaryotes, talking about the cell structures. I doubt we'll have time, but if we do, we'll get started on it. And then lastly, I'll talk about the endosymbiotic theory. All right, prokaryote cell size and shapes. Here we're looking at several different prokaryote cells. Their average size is really the average. It'd be 1 to 2 micrometers in diameter. They can be longer, like up to 10 micrometers in length. However, this is much less than 10 micrometers in width. So the diameter here is much less than 10 micrometers, but the length can be 10 micrometers long. Prokaryote cells tend to have three shapes: coccus, which are round cells; bacillus, which are rods; and rod and bacillus, or bacilli, are used synonymously; and then a spiral. So these are the three basic shapes that all prokaryotes have. There are different shapes, but many of them are based on one of these three. Like a coccobacillus is in between a coccus and a bacillus. Any question about any of that? All right, this slide is showing you different types of spirals. There is a vibrio, which is either a half spiral. There's a spiral there, spirillum. And a vibrio is either a half spiral, or you can call it a curved rod. Either way, curved rod or a half spiral, that is a vibrio. There are a few species that are actually vibrio. Let me see if I can remember one of them. It'll come to me. I don't remember. Um, there are spirals, a single being a spirillum. The spiral has a corkscrew type shape. It actually has a corkscrew-like motion too. And then there's a spiral with many different, I don't know, bends. And this would give a term spirochete. The spirochete, the spirochete is more flexible than the spirillum. Any question about any of that? Oh, here we go. The Vibrio is Vibrio cholera is a Vibrio. You don't need to know that, but I'm just giving you an example of that. Um, Syphilis is a spirochete. There are some prokaryote cells with unusual shapes, like the star-shaped Stella bacteria. This is an actual electron microscopic picture of the Stella, and this is an artist's rendition there. And then there's the square Haloarcula. It's an archaea, and actually I would call it a rectangle, not a square, because there's an actual electron microscopic image, and it's a little bit more of a rectangle than this one here. That's actually a rectangle too. Not quite a square, but it's squarish. So there are some bacteria with unusual shape, but by and large, most of them are either cocci or coccus or bacilli or bacillus or a spirilla, spirillum. So most of them are that. Most bacteria species are monomorphic, meaning the cells have a single shape, like right here, the stella, all are star shaped. Any question about any of that? Most bacterial species are monomorphic, they have a single shape. But a few species are pleomorphic, where the species takes on many different shapes. Let me blow this up a little bit. Here is a pleomorphic species, and you can see that one is almost cockeye. That one is almost a rod. That one is, I don't know, like a dumbbell. That one's kind of a, what we'd call that diamond type shape. Generally, if the cells are pleomorphic, it is because the cells do not have a cell wall. There are some exceptions, but many of the bacteria that lack a cell wall, they take on a pleomorphic pattern. Their shapes are pleomorphic, many different shapes. And there's only one family of bacteria which lacks a cell wall. The Mycoplasma. This is a family of bacteria that lack a cell wall. Any question about any of that? All right. So that's enough talking about the cell shape. Let me come down here. This is showing you a Coccobacillus. This is not a cellular growth arrangement. It shouldn't be in the slide. It is a shape where you have a rod, which you shorten it, like here, and you shorten it and you shorten it, and then it begins to become coccus. It's still somewhat bacillus, and that's why we call it Coccobacillus. And there you can see one that's just a little bit longer than a coccus. And this would be like a short rod. Oh, there's one that looks like a coccus right there, right here. The coccobacillus tend to be a little bit pleomorphic in that they can look like a coccus. They can look like a short rod, this one there. And then they can be definitely in between, like that one right there is definitely coccobacillus because it's not really a coccus, not really a bacillus. Any question about any of that? There we go. So let's talk about the prokaryote cell arrangement. This is oftentimes also called the growth arrangement of the cells. Different species can take different cell arrangements or growth arrangements. This is how the cells stick together. For the cocci, remember cocci is plural of coccus. For the cocci, you can have singlets, which this one here is a single cocci. That one there is a single cocci. You can have diplococci. That one there is a diplococci. This one here is a diplococci. You can have streptococci. This is where the cells are all stuck together and line up in a row. The cells are dividing on one plane here. That's why they grow only in a row for both diplococci and streptococci. If the cells can grow in two different planes, the cell, when it divides, can form a tetrad. And there's a tetrad there. That's the artist's rendition. You have to look very carefully here. And right there, 1, 2, 3, 4. There's a tetrad there. There are too many cells here, and so it's hard to see the cells there. The sarxinia is even worse because, for one thing, you can't see all the cells of the sarxinia. That's 8 cells. This is when the cells divide on 3 planes. They can form a sarxinia, and that's can be a clump of 8 cells stuck together. 1, 2, 3, 4, 5, 6, 7, and the 8th would be right here. You can't see it because it's being hidden by these 3 and maybe that cell as well. Let me blow this up and maybe we can see a sarxinia there. This 1, 2, 3, 4, 5, 6, 7. We can see 7 of 8 cells in this sarxinia there. There's another sarxinia there. A cocci can also form a staphylococci. That is where the cells clump together. If you put a stem on that, that would look like a cluster of grapes, and that's actually what staphylococci means. Cocci growing in a cluster like a grape cluster. So staphylococci actually means grape cluster, and then cocci, a ball type cell. And there you can see staphylococci there. So the cocci have the most cellular growth arrangements of all the cell types, meaning the different shapes. Cocci have the most cellular growth arrangements. For the bacilli, we can have a single bacillus called the singlet or single bacillus. And that would be a single bacillus there. This one there is a single bacillus. You can have a diplobacilli, and that's where 2 bacilli are linked together. But right there and right there is a diplobacilli. You can have a streptobacilli, and that's where it's the bacilli are linked together like a sausage link, meaning they're linked on the small side hooked to the small side. So they're running, I don't know, lengthwise to lengthwise. That's called streptobacilli. If they're connected on the long end, they are called a palisade. Let me shrink that down. And there we can see a palisade. Any question about any of that? Anyone remember from Mark Twain where Mark Twain was painting a palisade fence? What's going on here? Anyone remember that? Mark Twain was painting a palisade fence and he was trying to convince Huckleberry Finn that it was fun painting the palisade fence. So this would be the palisade fence where you have a board right there stuck into the ground right here. I'll make this wavy because this is the ground. And then the boards are just stuck one after the other. This fence used to be very common, not as common anymore. Oops, don't ask me what happened there. Ignore it, just bring that down. Um, Anyways, that's a palisade fence. And if the bacteria grow like that, we call it a palisade. Any question about any of that? Remember, a coccobacillus shouldn't be on this slide. It's not a cellular growth arrangement. It is a cell shape. And I got this slide from our textbook author, and I don't know why he hasn't corrected that. So the bacilli have fewer cellular growth arrangements or fewer cell arrangements than the cocci. And whenever you have a cellular growth arrangement, you will have more than one. Meaning if you have streptococci, the streptococci can break, and then you'll see a diplococci as well as streptococci. And the streptococci can break, and then you'll see singlets as well as diplococci and streptococci. Any question about any of that? And that's true for all of these. Whenever you have one cellular growth arrangement, you'll have more than one. So the cellular growth arrangement is pleomorphic with one exception, and that is that the cells only form singlets. You will only see singlets. And that's the only time the cellular growth arrangement is not pleomorphic. Any question about any of that? The spirals and the spirochetes only have singlets in their cellular growth arrangement. These cells, the spirals and the spirochetes, when they divide, they do not stick together. And so they only have singlets. Any question about any of that? If you see more than one cellular growth arrangement, you take the highest order and then you give the species that name. Meaning if you see singlets, diplococci, and streptococci, you say that species can form streptococci. If you see Sarxinia, tetrads, diplococci, and singlets, you say the species can form Sarxinia. Now, to get the cellular growth arrangement, you need to grow the cells in a liquid culture and then look at the cells directly under the microscope, usually in a wet mount, but you can stain them. The trouble is if you let the cells dry, or if you take the cells from a solid culture, like a petri dish, and then put it on the slide, you will get clumping to occur. So you can see Staphylococci and Sarxinia when they shouldn't be there. And then also if you are rough on setting up your cells, why we usually do it on a wet mount because it's very gentle, you can break apart the streptococci into diplococci. You can break apart the diplococci into singlets. You can break apart the staphylococci, although I'm not sure what that would break down into, but you can break it apart. And so you need to be very gentle and take the cells from a liquid culture to get the cellular growth arrangement. Any question about any of that? So on the gram stain, you generally cannot get the cellular growth arrangement because the gram stain is not gentle and it normally takes cells from an agar plate, and so the cells will clump together. And then it's also, the cells are allowed to air dry, and that will tend to clump the cells together as well. All right, let's talk about some of the typical structures found in a prokaryote cell. All the structures found in red are found in all prokaryote cells. They'll have the cytoplasm, they'll have the ribosomes, they'll have the plasma membrane, and then they'll have DNA. Actually, all cells have these four cell structures: the cytoplasm, the ribosomes, the plasma membrane, and the DNA. No, prokaryotes have a nucleoid region, which is a region of the cytoplasm containing DNA. Wherever the DNA floats in the cytoplasm, we call that the nucleoid region. It tends to stain differently than the cytoplasm, but there is no boundary between the nucleoid region and the cytoplasm. So where the DNA can easily just float over and move around in the cytoplasm. Any question about any of that? But all cells have cytoplasm, ribosomes, plasma membrane, and DNA, including eukaryote cells. Let's talk about the glycocalyx, shown oh, there it is here. This outer pink layer. The capsule is a glycocalyx. The glycocalyx is secreted by many prokaryotes, but not the majority. So majority of bacteria do not secrete a glycocalyx. And to tell you the truth, I don't know about archaea, how many secrete a glycocalyx. Obviously, some do, some do not. The glycocalyx is outside the cell wall. It's usually sticky, and there are three types of glycocalyxes. And this can be called glycocalyx as well as glycocalyx. Both pronunciations are correct. The three types of glycocalyxes are a capsule. It's neatly organized. There's molecules in it. It can help prevent phagocytosis. And you can see the capsule with the light microscope. There's the slime layer. It's unorganized. It's loosely attached to the cell, and it's mostly water. You cannot see the slime layer with a light microscope. To see the slime layer, you have to use an electron microscope. And that's because it's mostly water. There is a third type of glycocalyx. It's only found in biofilms. It is the secretion that actually is secreted by the bacteria, which then forms the biofilm, and it's called the extracellular polysaccharide. We won't talk much about biofilms here. I may briefly talk about it one other time, but we won't really talk about biofilms in this class. Any question about any of that? Of the glycocalyxes, we'll mostly talk about the capsule because it is the one you can see under the light microscope, and it's also medically important because it can help prevent phagocytosis. I suppose the biofilm is medically important as well, but we're not going to talk about why that might be the case. Um, the biofilm can help protect the cell as well as it puts the cell in different um, metabolic states, and so the cells may be more difficult to kill if it has a biofilm than if it's lacking a biofilm. I won't quiz you on the biofilm and anything about the biofilm. All right, let's talk about the cell structure of flagella. If a prokaryote cell can either move or swim, that means it has a flagella, because this is the only structure that prokaryote cells have for locomotion. The flagella is used for locomotion, and it will move in the opposite direction of the flagella, because the flagella, it sort of moves like a whip, and when it moves, it'll propel the cell this way. The flagella is largely outside the cell wall. It's made of the protein flagellin, just different chains of the protein flagellin. But there is a portion of the flagella which is actually inside the cell wall, this portion right here. And there's even a portion of the flagella which is inside the cell membrane, this portion right here. This is called the basal body of the flagella. You don't need to know that. But most of the flagella is outside the cell wall. But there is a little bit inside the cell wall and even in the cell membrane. If eukaryote cells can move, they can move by flagella, by cilia, and by pseudopods. But prokaryotes can only move by flagella. Flagella is the plural name. If there's one flagella, we call it a flagellum, singular. Any question about any of that? There are different arrangements that the bacterial flagella can take. You can have one flagella on one end, and it's called monotrichous. I won't quiz you on the flagella arrangements, but you do need to understand it for one of the labs. If you have flagella on both ends of the bacteria, it's called lophotrichous. Multiple flagella on both ends. If there's one flagella on both ends, it's called amphitrichous. And if there's flagella all over the cell, we call it peritrichous. With the flagella, when the cell first divides, it'll have no flagella. And then it'll put out one flagella, and so we'll call it monotrichous. And then it'll put one out on each end, and it'll be amphitrichous. And then multiple on each end, it can be lophotrichous. And then it can put flagella out all over the cell. So then it can be peritrichous, meaning when you see a species which is peritrichous in its flagella, you will see all the other types of flagella arrangements as well as no flagella. You can see that because the cell just hasn't made the flagella yet. But eventually that cell will become peritrichous if it's peritrichous. On the other hand, there's some cells that can only go up to lophotrichous, and it can have in the cell population, cells which are no flagella, cells which are monotrichous, cells which are amphitrichous, and then cells that are lophotrichous. For naming the cellular growth, the flagella arrangement, you name it by its highest flagella arrangement, meaning you don't say that the cells are no flagello, monotrichous, amphitrichous, and lophotrichous. You'd say that the cells are lophotrichous. Any question about any of that? And like I said, you need to know that for one of the labs. You don't really need to know it for the quizzes. But if the cell can rotate, that means the prokaryote has a flagella, allowing the cell to either run or tumble. This can give the cell movement, which is taxis. So taxis is movement toward or away from a stimuli. Here we have a light source, and the cells are actually moving this way, meaning they're moving away from the light. That means these cells are not photosynthetic because they would move towards the light if they're photosynthetic. There are two different types of taxis. There is chemotaxis and phototaxis. Phototaxis, I've already talked about, is where the cells either move towards or away from light. Chemotaxis is where the cells either move toward or away from a chemical. And so if the cells are moving away from the chemical, they'll move down the chemical gradient, meaning to a region where the chemical is less concentrated. Any question about any of that? All right, let's talk about the endoflagella. Axial filaments are also called endoflagella. This is a flagella that is different because the flagella is found inside the cell wall. Let me blow this up so you can see it better. There is the spirochete, and there's the cell wall. Let me blow that up even further. The pink is the cell wall, and the axial filaments are surrounded by the cell wall. It looks like in this graph, it's the outer sheath of the cell wall, which is surrounding the axial filaments. The point is the endoflagella is in the cell wall, and it wraps around the cell. Let me shrink that down. Like this, wrapping around like that. And then when the flagella moves, it gives the spirochete a corkscrew-like movement. The endoflagella is only found in spirochetes, and it is anchored at one end of the cell. Well, it looks like we're going to have a commercial, but let's see.
I can never remember how to perfectly format my.
When it moves, the flagella doesn't whip like a whip, causing the bacteria to move this way. It causes the bacteria to move in a spiral type movement like that. And here's something you notice how flexible the spirochete is. Yep, I didn't go back far enough. Here it's moving more or less north and south, and you see how it just bent, and now it's moving east and west. When the spirochete moves, it will cause the thing to move in a spiral, and it's moving constantly this way. If it were to change its movement, it could then move that way. Any question about any of that? We've got something else open here. Let me shut that down. It's thought that this corkscrew movement can allow the spirochete to actually burrow through a mucous membrane. And that's why when you get a spirochete infection, you can actually burrow through the mucous membrane, which allows it to then infect a patient very easily. Any question about any of that? Actually, it's after 6 o'clock. I thought I would allow you to have a longer break today. Are there any questions? If there's no questions, I'll see you in the lab at 6:30.
Dr. Volpe, I was looking for the infectious disease project instructions, and I'm not finding it on my end.
Okay. Do I have the share? No, I don't. Let me show you. It's in the Canvas webpage. I think it's lab 2.
That's why I'm not finding it. It says to look at project instructions, and I just couldn't find that.
You're talking about the infectious disease research
Yeah.
project? Yeah,
Yeah.
it's right here under lab 2.
Okay, and then the instructions are just in the document in the introduction, or what
You
instruction?
should also see the lab where I discussed the infectious disease research project.
Okay, and that's on your YouTube, you said?
It's on my YouTube website. Um,
Is it just
I need to go up a little further. Um, so it should be in week 2. Probably it'll be on Tuesday of week 2.
okay, perfect.
October
Yeah.
2nd or 1st, probably 1st, October 1st of week 2. So you should see the lab where I talk about the infectious disease research project and then come up with a topic. Let me mention this for the other students. Am I still recording? I think I am. You do need to get your infectious disease project. You need to clear it with me this late in the game. Please submit more than one possibility because uh, students are only allowed to, the first student who asks gets that infectious disease. And so the next student coming along will not get it. So if you ask for like 3 at once, you're much more likely to have a infectious disease that nobody has asked for.
Oh, okay, gotcha. And that
Right.
will be in the YouTube video. All the
All
ones
that
that we
is
choose
discussed
from.
in the YouTube video.
Okay, perfect.
All right.
Thank you.
All right. Bye.
Bye.